June 1944, Normandy beaches, D-Day +3. A British medic crouches in a foxhole, watching a soldier die from an infected leg wound. The injury itself wasn't fatal, just shrapnel. But infection is spreading, and the medic has no penicillin left. 30 meters away, an American medic treats a similar wound with a small vial of yellowish powder. That soldier will survive. The British discovered penicillin. The Americans figured out how to make enough of it to actually save lives. September 1928, Alexander Fleming returns from vacation to find mold growing in his petri dishes at St. Mary's Hospital, London. The bacteria around the mold are dead. He's discovered penicillin, the first true antibiotic. 1929 to 1938. Fleming publishes his findings. Nobody cares. Growing enough penicillin to treat even one patient requires months of laboratory work. The yields are microscopic. The process is impossibly inefficient. It's a laboratory curiosity, nothing more. 1939. War breaks out. Bacterial infections kill more wounded soldiers than bullets. Doctors desperately need antibiotics. Fleming's discovery sits unused because nobody knows how to produce it at scale. 1940, Howard Florey and Ernst Boris Chain at Oxford University read Fleming's old papers and see potential. They begin experimenting with growing penicillin using available equipment: bedpans, bathtubs, milk churns, anything that holds liquid. After 6 months, they've produced enough penicillin to treat one patient. Albert Alexander, a policeman dying from a scratched rose thorn that became infected. They inject their entire supply. He begins recovering immediately. It's a miracle. Then their penicillin runs out. Alexander dies. Florey and Chain realize the horrible truth. They've proven penicillin works, but they can't make enough to save anyone. Their laboratory methods produce maybe 10 doses per month. The war needs millions. British pharmaceutical companies examine their process and decline to help. Britain's factories are building planes and tanks. There's no industrial capacity for experimental drug production, no expertise in fermentation at scale, no time to develop new techniques while German bombers target London every night. June 1941. Florey makes a controversial decision. He'll take his research to America. British scientists are furious. This is a British discovery. British universities developed it. Now Florey wants to hand it to the Americans. Some call it betrayal. Florey sees it differently. A drug that can't be produced might as well not exist. Pride won't save soldiers bleeding out in field hospitals. He arrives in America carrying vials of penicillin mold smeared inside his coat lining, hidden in case German U-boats sink his ship. If he dies, maybe the mold will survive, and someone will figure out what it is. He meets with the U.S. Department of Agriculture in Peoria, Illinois. Not a pharmaceutical company, not a university. The Department of Agriculture, because they have the world's leading experts in something Britain never prioritized: industrial fermentation. The Peoria facility spent decades perfecting large-scale fermentation for producing alcohol during Prohibition, then pivoted to industrial solvents and food processing. They know how to grow things in giant vats efficiently. Within weeks, American researchers make the first breakthrough, switching from growing penicillin in shallow containers to deep tank fermentation. British methods used hundreds of small flasks. American methods used 10,000-gallon tanks. Then Mary Hunt, a laboratory assistant, brings in a moldy cantaloupe she found at a Peoria fruit market. The mold strain from that cantaloupe produces 200 times more penicillin than Fleming's original sample. But The real breakthrough is philosophy. British researchers approached penicillin as a scientific problem requiring laboratory precision. American researchers approached it as a manufacturing problem requiring industrial efficiency. They didn't need perfect sterility, just good enough. They didn't need pharmaceutical-grade equipment, just food processing tanks. They didn't need to understand why it worked, just how to make more of it, faster. 1942, American pharmaceutical companies, Pfizer, Merck, Squibb, begin converting factories to penicillin production. They're using deep tank fermentation techniques borrowed from beer brewing, quality control methods from food canning, and packaging systems from consumer products. The approach horrifies British pharmaceutical traditionalists. This isn't proper drug manufacturing. Where's the precision? The scientific rigor? The careful quality control? But it works. 1943 production, 21 billion units of penicillin, enough to treat thousands of soldiers. 1944 production, 1.6 trillion units, enough for every wounded Allied soldier. By D-Day, American factories produce more penicillin in 1 month than all British laboratories combined produced in the entire war. American medics carry penicillin as standard equipment. British medics request it through supply channels and often receive nothing. The infection mortality rate for American wounded drops from 18% to 1%. The infection mortality rate for British wounded remains above 10% through 1944. Fleming, Florey, and Chain received the Nobel Prize in 1945 for discovering and developing penicillin. The British press celebrates it as a triumph of British science. The American factory workers who actually figured out how to produce it in quantities that saved hundreds of thousands of lives received no recognition. Their names don't appear in medical textbooks. Industrial fermentation specialists don't win Nobel Prizes. Meanwhile, British pharmaceutical companies that refused to help Florey in 1940 now scramble to license American production methods, paying royalties to use techniques for manufacturing a drug Britain discovered. Fleming discovered penicillin. Florey proved it worked. But American industrial engineers saved the lives. Sometimes the breakthrough isn't the discovery. It's figuring out how to make enough of it to actually matter. Science discovers. Engineering delivers.